Welcome to the deep dive. Today we are jumping right into a really fascinating new microscopy technique. It's called bidirectional quantitative scattering microscopy.
Or BiQSM for short.
Right, BiQSM. And our mission today is to figure out how this one innovation might finally solve one of the biggest trade-offs in live cell imaging.
Oh, it's a classic problem. It really is. When you're trying to image cells without labels, you've always had to make a choice.
A choice based on what you want to see, basically.
Exactly. On one hand, you have Quantitative Phase Microscopy, or QPM. It uses what we call forward scattering, or FS.
And that's the light that mostly goes straight through the sample.
That's the one. It's fantastic for the big picture. The larger microscale things like the cell's outline, big lipid droplets, we're talking scattering here.
But because it's so good at the big, slow stuff, it completely misses the tiny, fast-moving particles inside the cell.
Totally misses them. The nanoscale action is invisible. So to see that, you'd have to switch to a different method.
Which would be iSCAT, interferometric scattering.
Right, and iSCAT uses backward scattering, or BS. That's the light that bounces straight back.
Yeah.
It's incredibly sensitive to tiny things, objects smaller than 100 nanometers.
So it can catch those little 30-nanometer beads zipping around.
It can, but you get almost no context. You see the fast-moving dots, but you lose the overall cellular structure. So it is always high sensitivity or a comprehensive picture, never both.
Until BiQSM. The whole idea here is that it ends that rivalry by integrating both FS and BS detection at the same time.
Simultaneously, using a single objective lens and a single image sensor.
Okay, but hold on. Capturing light going in two opposite directions, forward and back, on one sensor. That sounds like an alignment nightmare. Do they have to sacrifice clarity?
You'd think so, but not at all. Actually, the beauty of using that single setup is that it guarantees spatiotemporal consistency.
Meaning you get two different images of the exact same spot at the exact same time.
Precisely. And the immediate result is, well, it's dramatic. You get a dynamic range that's 14 times wider than QPM could ever achieve on its own.
14 times. So you can literally see the slow, big structures right alongside the hyper-fast nanoscale particles.
It's the same frame.
Okay, so achieving that range is one thing, but I imagine interpreting that flood of new data is the real challenge. And that's where the correlation analysis comes in, right?
That's the real breakthrough. It allows for an enhanced particle characterization that was just impossible before in label-free imaging.
What do you mean by that?
Well, with the older methods, things like size and refractive index would kind of blur together. It was hard to say what a particle actually was. For instance, FS analysis alone couldn't tell the difference between a 150 nanometer polystyrene bead and a 200 nanometer silica bead.
They just look too similar.
Right. But with BiQSM's correlated data, you can easily differentiate them. You can decouple those properties, size, density, refractive index. It's a huge deal for understanding what's actually inside a living cell.
And we really see the power of that when they started looking at the cellular dying process.
Yes. The time-lapse observations showed two really distinct stages in how things moved inside the cell as it was dying.
Wait, the cell's internal chaos changes in organized phases? That feels counterintuitive. What was stage one?
Stage one is what they call the activation stage. You see this transient activation of the microscale particles. They start moving more. And that coincided perfectly with a 50% jump in high-frequency DS signals.
High-frequency BS signals. So that means the tiny nanoscale objects are all of a sudden jiggling and fluctuating much more rapidly.
Like a burst of frantic energy.
Yeah.
And then you hit stage two.
The attenuation stage, where everything slows down.
Exactly. The microscale motion gets damped, and this is the crucial part. The low-frequency BS signals drop by about 50%.
And that's the slow collective flow of the nanoscale objects.
And it drops specifically in the nucleus and other regions that looked empty. That's synchronization. A 50% increase followed by a 50% drop. It's almost like watching a cellular heartbeat stop in two distinct phases.
So to wrap this up for you, what BiQSM really does is bridge that critical gap. It captures a complete simultaneous picture of both the cell's structure and its internal dynamics.
It gives us a unified way to watch a cell truly live and then truly die.
And what's really fascinating here is the potential.
I mean, if these motion changes are so precise during cellular death, they could be standardized. They could become diagnostic indicators for the early detection of cellular distress.
Imagine that. Tracking the health of a cell not by which chemicals it absorbs, but just by watching the change in its internal jiggle. That could revolutionize how we think about cell viability forever.